to your department, give a seminar about my current research. And one thing I didn't mention, my daily job is not doing research. It's for checking the sample, whether you like our daily food is good or not, and before reach the customer like everyone. So my director very support me, so I can have almost 20% of my time doing research and to update the current message, and that's why I'm doing over here. And today my seminar we will focus, we use the combined two technique. One is the fluorescent microscope and the technique, and another probe specifically for chitin to detect the insect fragment for filth analysis. And begin talking and work at, I will tell you why we need to do this kind of work. So our daily food, most time we contaminated by insect. We all entomologists, so we know it's almost impossible our daily food is free of insect. Also that our food, when you in storage process, also can contaminate by small animals like a rat, mouse, that's the bird, and other kind of small animals. So we all know that for the whole insect identification, most time we have all different kind of K, like antenna, leg, eyes, elytra, all that stuff we can use to identify the insect. But if the insect in your food, like bread, when going through that process, already become fragment. So it's hard to see, but it is there. So first step, we need to extract that filth element from our food sample. And we most of the time we digest them and wash them over a sieve and left a smaller amount food residue with all that filth elements in there. That's a plate. So then next step will be under microscope, we recognize and separate and counting all the insect fragment and with a noise food residue background. Then so we can say that elytra and some hair from small animals and mandible from part of the insect. So FDA regulate our food. We all know that's impossible, all the food free of the insect. So FDA have a defect action level. It means provide a guideline for food process on a level. So it's, if certain amount of insect fragment in the food will be okay. If over the threshold, then will be a violation. So FDA will take action on that. So we all know the different insect can have significant different effect your food quality. Like this one, it's copper beetle. It's a very strong storage pest and can chew in and eat almost everything. Even your packaging material can burn through. So that's significantly affect your food quality. And also insect also can carry the pathogen that can cause the problem. And also some insect is a quarantine insect because in United States, we never have that kind of insect. But it will be if the food import from another country come to US, if alive, maybe can start a colony. So they cause extra problem. That's why we need to pay attention to the insect there. Okay, yeah, next slide, I will show you a more detail how we, as a field analyst, as an entomologist in the FDA, what we do. We already know that Petri dish has a filter paper, carry out food residue and insect fragment. So we under microscope, we manually separate the food residue and the insect fragment. So you can see on this pictures, we have food residue over here and over 1,000 insect fragment over there. But the current that we manually separate the insect fragment under microscope with a noise background is very challenging, also time consuming. Also, the results in inconsistency because you can see the color, its texture. Sometimes the food skin, like seed skin and fruit skin, it's very similar with the insect fragment. It's if you don't have any experience, probably you cannot separate them. And have more experience, you will more recognize which part is the insect fragment and which part not. So that expertise is also very difficult to transfer from one person to another person. Then species ID it's also will be very challenging. I think most graduate students, when you go to taxonomic of the insect, you will notice, okay, some species is pretty easy, but some species it's you need to dissect them and pay close attention. So that's also a challenge, especially we only get part of the insect. So you don't have a whole specimen for you to examine that. So that's a problem. That I show you some case like FDA sample number, and there's our system number and private lab number, because the food quality also checked by the private lab. So FDA can audit them results. Like a private lab and find these three, you can see 800, 1100 insect fragment. When we're auditing them results, a same product from same lot, we only find much less insect fragment. So it means like over here, that's them mistaking this one as insect fragment because this is a seed skin. It very similar there. But another kind of error can make, like this private lab only found 21 insect fragment. But when we go to FDA lab to auditing, we find almost 10 times, 243. So that's kind of two-way error. If you don't have enough experience, you can make that mistake. So most of that probably is the appearance of one object can be significantly changed and affected by a lot of factors, go through the food process. But if the components for the object, it might be not change much. So that's our CART project will follow this principle to identify the object as an insect fragment by targeting a very specific compound, chitin. At the same time, this method does not change the appearance of object because if we find an insect fragment, we think it's a violation, we need to keep that evidence. If the private company said, oh no, we need to challenge them. So we need to show you that evidence in our hand. So that's why, so next question will be why we choose chitin. So as an entomologist, that's an insect, we can go to different part. So chitin is one most abundant biopolymer in the nature. Cellulose is number one, chitin number two. It's widely distributed abound like insect arthropods, insect and spider, nematodes and fungi, but chitin is absent in vertebrates and plants. You know, this vertebrate and plant is the most ingredient for daily food. So for the insect, we can go to all different parts. Everybody probably already know, except the middle gut on this part, no chitin in there. All other parts all have chitin. So that makes the chitin a very good target chemical for we tax that if we can mark a, a kiting, target kiting, and then we can easily identify that object is insect fragment. So our short-term goal will be mark insect fragment and separate them out from food residues. So people maybe ask why you don't use the traditional chemical dye, like you go to tissue and dye that by blue and green, because that will be change the morphology of character of the insect fragment. So you cannot identify them under normal light system. So this new technology will be mark the kiting, and under microscope, normal light microscope, it's just look like the same as before, but under fluorescent light system will be different. And also counting and measuring insect fragment automatically under microscope. We have a microscope have the capability can scan the whole petri dish line by line and have the labeling them which point have the interesting object, and then change it become normal light and zoom in, take the pictures, and then will be identified that. Our long-term goal will be developing an automatic plate reader by combine molecular mark technology with artificial intelligence, AI technology, and also develop a filth elements identification system by image analysis and pattern recognition, including all kinds of insect fragment, hair, glasses, and metal. And then we combine number one and number two, building an open automatic filth analysis system that has self-learning capability. That's the AI technology involved over there. So let's say that the first technology we use as a kiting probe. And then we go to the very basic chitinous is some kind of enzyme combining and degrade chitin. That's in nature, it's in there. And a lot of research on that chitinous enzyme for developing pesticides. It can be labeling with fluorescent dye, such as green or red fluorescent protein. It's already widely used as biomarker. The chitinous is combining and degrade, but the CBD chitin binding domain is part of that enzyme, referred as a portion of protein less than 70 amino acids on this part. And only it still have high capability to binding chitin, but not degrade the chitin. So then we can put that fluorescent protein with this part and make a probe. And a chitin probe, CBD proportion as 70 amino acids and plus fluorescent dye. And then as will be still have strong capability binding with the chitin, but not degrade the chitin. Then we make the probe. And this kind of technology have the advantage to compare with our current approach. All possible insect fragment, adult and larval, can stand out for recognition over black background. And current method for adults, we can say leg, antennae, and elytra is no problem. But for the larval, it's hard to find that. And also this kind of technology did not destroy or hiding any morphological character that normally used for identification. And also all plant material will not interfere with the element analysis, with the filth element analysis, and also provide a base for final goal automatically plate reader and identification system. And this knowledge limitation will be does not including all other filth elements such as hair, glasses, but it's, with our work, we just found out it also working with rat, mouse hair. Whether it working for the human hair, we highly expect it will work, but we not try yet. So later on, I will be explain why it works. A challenge, you know, every day we have eat so many different food. So a matrix will be food matrix with a big challenge, and different food matrix may be affect. From principle, we know it doesn't affect it, but we need to try that. Also, fungus might be in our daily food. That fungus cell do have the chitin. So how to eliminate this challenge? So that's our uh, working try to solve. So I gave you a progress report, and this we man-made electrode. We cut the electrode become square, and these are normal lights. So you can see it's a adult beetle electrode, and we treat that electrode by green dye, green chitin probe. You can see that only broken area become green, and also broken area and some point. And we know that chitin probe can label the insect fragment. And also if we let them go through acids, that's normally with digestion food, and destroy the layer on the surface. So you can see the whole electrode is labeled by the chitin probe. This is red dye. So I use the green dye and red dye, and just limit like a traffic light. This green light, this red light, this yellow light. That's normal color over there. So we just talk that this adults, and we talk the larva. This one, several ham beetle larvae we found from our sample. So this all the larvae is treated by chitin probe. And you see on the normal light, it just look like regular larva, different larva. These two treated, this one not. And under fluorescent microscope, you can see these two significant labeling by red dye. And that one is just on the background for this one. It's not labeling. So it means that dye really work, also have specifically for insect. And this one I will show you same kind larva. It's treated by green dye. You can see that's normal light, that's larva, that's head and the whole larva over here. And after under fluorescent light system, you can see the whole larva, it's green. And over here, normal light, it's the head capsule, it's broken. Over here, you not see it very clear. But after the mark, because that will be the layer, the chitin will be exposed. So combined with the probe, it's much clearer over here. So that's, so we're showing that probe is working. So I will try the different insect. So that's the aphids. There's the mouse larva head. That's a leg. That's a shrimp abdomen. Has different color. It's a green dye and red dye. That's all this man-made elytra, adult beetle elytra with the hair. You can see the red hair. That's red rodent and mouse hair. So we can make it green, red. And we also can make the hair, if we use the green dyes, you can see the hair become green. So that means that we can add one more filth element in there. So for our regular plates, we treated plates, used our red dye. So under normal light system, you can see all different kind of fragment like elytra and larval and adult leg antenna. Over there, there's a larva. If we move that picture is going to under fluorescent light microscope. You can see like this. Background is dark. All that insect fragment is stand out. So we will go see the part. After this part will be over here. You see one elytral, one larva over here. This one will be adult antennae over here. This leg will be here. This part will be this part. This larva will be this one, and this one will be over here. So it means this whole thing, if you go to a big plate, it's working. So then before we know that proper design for the chitin, specifically chitin, and why it will also work on the hair. So the chitin and the cellulose, you can see only difference over here is the carbon and the carbon a bond. It's for cellulose over here. But for chitin, it's a carbon to nitrogen, this bond. So let's work on this part. For the hair, have this kind of chemicals, also have carbon and nitrogen bond. So that's probably the possible mechanism is there, but we're not sure. So that's just, we think it's possible there. And over here, I show you another 3D dimension for the chitinase enzyme. There's the whole thing is a chitinase enzyme. And CBD on this part, CBD on the green and the lemon color, just this part. That's the 70 amino acid part of the enzyme. And have that capability bonding with chitin and fluorescent dye and not digested, that's chitin. That's what we use over here, this part. So far we know as a filth analyst or entomologist in FDA, we extracted the filth elements, we recognize, separate, and counting the filth elements. And next part, we need to identify that. So we use the chitin probe. We can recognize, separate, and count it, but how we identify the insect fragment. So next step, I will be talking this part. And so far we already know your fingerprint, your face can use like identify a person or object that QR code. It's working that way. So we think a literal like this one, insect fragment, whether it can acting as a fingerprint for this species. So we can use part of that a character to identify the whole insect. That's the idea in there. So we select 15 different storage paths, and some at a different family level, and that like have a same family, and also same genus, and different species. That's all 15 different very common storage paths. So we collect all of them and take the elytra and take the image. So, and we use the image analysis, extracting the meaningful information from highly qualified digital camera image to get the information, and also use pattern recognition. We all know on the elytra have a different pattern, and the algorithm can perform most likely match of the input and provide a reasonable answer for all possible input. So then will be advantage for this technology consistent with the scientific method, so the other can produce and validate one other person's result. Because in FDA, if we find that this product is validated, and the first analyst finding needed a second analyst to confirm that. And this technology can make it automatic process and a large amount of data. fast and precise, and low cost rather training human beings. That's we going there. So far we already developed a prototype system and can work for the whole electrode. This system only have two windows. First window asks load your image, and you load the image, and then said, okay, go through that identification. That bar will go through, and the computer will match your image going to the database, and the algorithm will calculate most like, and then give you a result. That's load your own image. That's a database. Then will give you let's have 99% confidence that this is species A or species B, and we have the threshold. If lower than 90, we don't trust that just as a reference, and we go through and we identify them. That system is an open system, so if we confirm that identification, we will enter a system. The system will be remembered that, okay, this one is species A, and that's the AI technology involved. So the accuracy by the species, and we use 8 different neural network model to train the whole system. The accuracy will be 89 to 90% accuracy. Okay, some species it's 100%, but some species are lower because that's just the same genus. It's look like from naked eyes almost identical. So computer also will be confused by that. So, and so far we have several papers already published. One is our image analysis environment, that basically interface how to use that. And another one is species identification, user image and pattern recognition. And another one will deep learning, so AI technology involved there. And we all know the computer is not human brain. And if you input a good image, will come out a good result. So we think that's how to take a high resolution, good image for the system. We develop a SOP for that. And we also compare different neural network, which model working better. That's all this explore research going on. And this part is done by our NCTR, as a bioinformatical person that's over there. I have a computer scientist working with the algorithm and AI technology. So, very beginning, I tell you as an entomologist in the FDA what we do, but that's a current approach. But in the future approach system will be start on that's a plates. The plates are treated by a CBD probe, and then going through a microscope and have a scanning, image taking, and a counting system. So that system will be a counting and a measurement and take an image. Then the intelligent recognition system will take as raw data, and doing all the algorithm will do the calibration, similarity measurement, become identification results. So we have that confidence at identification. This system will be accept more data and real time. It's just an open system that will increase the capacity and accuracy with a more authentic image add. This system will have self-learning capability. And we all know that right now AI technology, if you put more authentic identification, the system will become smart and smart. So that's our future goal. So thanks for Arkansas lab, Amy Bourne is my boss, and Kanika and my co-worker, Hamancho, is our branch director, and he allowed me accept my daily work and have some extra time to doing the research. Also, our research work with FDA, another lab at the Atlanta lab, and David and Cherry, and it is supported by Office of Regulatory of Science, Food and Drug Administration. So that's all my talk. If you have any questions, just free ask me. I will try my best to answer as possible. Thank you.
Thank you. All right, we do the online into the reaction. There we go. And so what I there's lots of people online. Go ahead and ask questions. The one thing that is usually helpful is if you raise your hand. So in the reactions, there is a raise hand option, and then Dr. Ding can then call on you to ask the question.
So I can end the slideshow, right?
Sure. Yep.
Yes.
Right, there's some sort of chat going on, but go ahead and ask your questions, and I see Michael.
Yeah, it looks like
Can
Michael's got a question.
Can you see the hand risen, or do you want me to call people?
I think you call the people, so okay, yeah, you call the people. I listen to the questions, so I make sure I understand the questions. Yeah.
Michael, go ahead. You're muted. There we go.
Hi, my name is Michael. I'm a graduate student in Dr. Hamilton's lab. Fascinating talk. I actually do work with archaeological specimens of insects, which are a lot of times almost always fragmented and really hard to identify. And I was wondering if you thought that that staining technique would work on specimens that were really old or degraded, like where the chitin, is still intact but like starting to break down. And then further, like if that were applied to them, like, do you think that those specimens would be destroyed or like ruined afterwards?
Okay, yeah, I think you have your question is several part. I will try to answer one by one. First of all, I think you said your sample specimen that are old. I think that old, probably the chitin it's pretty stable, so should be work. Like our specimen, we go through the food process, the heating, all the process, also go to acid. We need the digestion of the food. It destroys a lot of things, but the chitin is pretty stable. Should be works. And also image analysis, like if you just like for it's you don't destroy your sample, you just take the high qualified, high resolution image, so it should be fine. I saw some literature about right now in FDA lab in Urban, and some people try to work on fly wings. You know that fly is a big vector for human beings. So we sometimes will find that adult fly wing. So part of the wing, we want to know whether we use the image of the wing can identify the fly. It started to look like promising, but not completely yet. And I know one reference, and I'm working on dragonflies. Dragonfly that wing, it have a lot of cell over there. So it's definitely working for dragonflies. I'm not sure I answer all your questions. If not, you can ask, because before that, the sound, it looked like a broken sound. Some word I'm not heard very clear.
You did answer all the parts of my question. Thank you very, very much. I appreciate it.
No problem. Yeah, if you need more information, you just send me an email. I can tell you on all my progress and let you know. Yeah.
Oh, I will be.
Thank you.
I will be following up with an email, definitely. Thank you.
You're welcome.
Shodan, go ahead.
Hi, Dr. Ding. This is Shodan from Dr. Chang Wang's lab. I have
Okay.
question. Yeah, I have a question. So if there are many kind of species and they are from different orders, like you have both moths and also the beetles, can you still identify them with your methods? And also my second question is, you talk about the to ID the electron, and how about if there are so many larvae instead of the endosphere, can you still ID them with this method? Thank you.
Okay. Yeah. I think like for that should be we try that first. Why we will try to use adults beetles because that's the common we found, and so we try that. Then later on we put because in our daily food we found so many different insects, and we tried aphids as the agricultural pest, and syrup, and like aphid syrup and other kind of like moss, different moss. Indian meal moss is also common we found, and very beginning we have 15 species storage pest. Right now we expand to go to 50. That's can cover our daily specimens almost 95%. So and also flies because on a cockroach, they all it's working for them. So I collect almost at least about over 100 species. We from our daily work, we collect all that. If we find any food, I will ask them keep that, and I use the kiting probe. It's working. So basically, every species should be work, but I cannot give you 100% confidence because I never tried some, so I only tried right so far about 100 species. Yeah.
There's so many, already a lot, 100. How about the larvae? I mean, the
larvae, no problem. You can see I show you the red leg beetle larva. It's working. Actually, on the larva, it's working even better because larva it's much softer, and on the skin layer, like a elytra, on the layer, we know inside the skin and have a different layer. So on the larva, it's working. It's better. It's more the chitin mark is much stronger, and even that like all the inside organ, except for the middle gut and other part, you can see pretty clear. And after aspiration tube, you can see that all is there. It's I'm working on for fly larva, and it's working pretty good. Yeah.
Okay, thank you. Thank you so much.
No problem. Yeah, my pleasure.
I think you can go, Dr. Hamilton. I don't think Dr. Ding can see the hand, so I'm just calling.
Yeah, I saw Dr. Hamilton have a rich hand. Yeah, okay, but he's mute. Yeah. Dr. Hamilton, you need to unmute yourself.
Yep, there we go. Thank you. So my question is about the imaging system that's being put together. Is there going to be a need for verification? I'm assuming that this is supposed to be set up as an autonomous system and just do everything, but is there still, I guess the basis of the question is this viewed as something that's going to take the human component out of the identification process? I guess is what I'm trying to ask.
Yeah, for current, we're doing that before we collect that image, take the image, all the 15 species we go through analysis 1, analysis 2 is all the authentic specimens. That we want to input it right. And after that, go through that computer system. Basically, that one electron, the computer system will cut it at different part, become 1,000 pieces fragment. Then make the pattern, the calculation, and come out, compare with another part, also authentic specimen. So we take the image. That image never going to the system before. It's called we call it training set and testing site. Like we have 30 specimens and 15 for training system, another 15 for test system. So we come out at 90% or 95% accuracy. And this system also the open system, like every unknown specimen we put in there, because you only have a 15 as a database, will come out, okay, a species A, on accuracy only 10 or 20%, because it's definitely not that species. Then we will be tell the system, okay, this authentic identification is species C. So we'll be enter the system. A system and a database become bigger. And if next time another kind of specimen come in, then that whole system will become 16 species. So we become bigger and bigger and smarter and smarter. So all that information must be confirmed by human eyes currently. But we want to eventually, if we are database big enough, and for the common species, like right now, we are 15 species expanded to 50. That's on the list. We will not need human confirm, because the system we already confirmed for this 50 species. It's good enough. That's the current situation.
Thank you.
So this is Dina, and I work on mosquitoes and ticks. So I'm not particularly familiar with, what kind of insects are found. But you mentioned I work with invasive species. I'm actually keeping my video out because there's some like a lot of sort of lag in the video. So when you discover a species of, say, that you can't identify, what do you do with that information? I mean, you mentioned invasive species. What do you do if you can't identify something? And what's the level at which you say, I can't identify it because of the quality of the sample, or I can't identify it because it's something new?
Yes, like most of the time we approach like that. If our manual asks you identify them as low as possible as your confidence level. If your confidence go to species level, we will go to species. If you are not confident with your identification, and we go to genus. If you're still not confident, we go to family level. That's for our current situation. And then later on, at some fragment, we just cannot. We are sure it's insect fragment, and we just say unknown. That's our current approach. So for our daily sample, we just do some analysis. Okay, I'm sure that's Tribodium. Some species go to genus. Some analysis, no, I'm not sure. I just go to family level. And the second analyst can come in and say, okay, I'm sure I can go to genus level, even species level. And we find a lot of mandible. and storage past and for the larval mandible, adult mandible, we have a pretty good collection, so we can easily go into a space level if you are experienced and after training. That's what we do. For the system right now, for we building a system, we only right now we have the adult electro for 15 to 50 species. We'll have a confidence. For mandible, we started working on take the image and building because that's with mandible we want to take the 3D dimensional pictures, and that will be different algorithm. So we try to find some new computer scientists help us on that part.
So, but you you are including species that are not presently in the United States, things that you're worried about?
Yes.
Okay.
Yes, like copper beetles, we include the copper beetle in that system. Because copper beetle so far, officially, U.S. said we don't have a copper beetles.
Wonderful.
But
okay.
we do know one time, like 10 or 15 years ago in California on the port, have the infestation, live infestation there. But the U.S. government put a couple $1 million to get rid of that problem, destroy everything around there. So far we caught it quarantine insect, and USDA pay a lot of attention, said no matter live or dead, we want them. So that's the situation. If we find a part of that copper beetle like well lab last year or 2 years ago, we found some. Then we identified the copper beetles. USDA immediately sent people come to our lab, said, where is the sample? We need to destroy the sample. And the specimen, we need to take it. And then going to molecular biology, gene expression, PCR, to confirm that the copper beetles. So
Great.
right now, if that, yeah.
No, that was that. You answered my next question, so we're good.
Okay.
Thank
you. Yeah.
So since we have like I mentioned earlier, we have a group of students that are taking the class. But since we have some time and I'm not seeing any hands raised, I welcome the students to ask their questions a little earlier than we normally would do it. So I'm seeing Jacqueline.
My name is Jacqueline Johnston. I am an undergrad. My question was, where would these systems be used? Would they be used in places where the food is being produced, or would the FDA inspectors only use these systems?
Yeah, that's currently the system was developed by us, and we only use for FDA part. Because the system not a verification yet, we need like a single lab validation, multi-lab validation. After validation, if private lab want to use it, that's fine. I do get from an industry from Florida. I'm saying, oh yeah, this system, can we use that? And we said, not yet. We'll be waiting when the system become much more mature. We have more confidence. We have more species in there. Yeah, so far we're only used by FDA lab. Yeah.
Thank you.
Thank you.
Jim has his hand up.
Yes. Hi, Dr. Ding. I may have missed this, but there's a lot of food produced in this country. What % of the food gets screened, and is it screened by the manufacturer, or does the FDA do it all?
Yeah, that's pretty big questions. Tell you the truth, probably you don't believe. We only screen about 2 to 5%.
Wow.
But we do have a pretty good system for targeting. And if like one food from other countries and try to get into U.S. market.
Mhm.
Then first you need to register. And your FDA right currently will be send, sometime we send an inspector, we call customer safety officer, go to that country, examine that manufacturer whether and GMO, good manufacturer practice while I'm doing that. And
Okay.
then we check them. When the food come in, if good, then we be give it a green light probably for a couple of years. We never check that. If
Ah,
we first time check that it's bad, then we put on that them on a list. It's like a blacklist. I said, okay, this manufacturer not good. Next time coming, we will check them again. If second time it's still not good, then we will put that automatically detention. So if the third time coming, we not even then want to check that. You need to hire a private lab to check it, and the private lab get a result, send to the FDA. FDA review it, said, oh, it's fine, and we will come back. If FDA said no, not good, then it's still not good. So we
uh-huh.
even not check them. Yeah, that's the protocol we're doing.
All right. Thank you.
Thank you.
So if you don't mind, I have a follow-up on that. I mean, this is Dina.
Mhm.
Because of and you obviously you're optimizing this approach. You're trying to make it so that it's more sensitive. And are you going to have to change your thresholds for deciding if something is bad or good, or rejection or acceptance of a food item?
Yeah, we have a called a defect action level. If we have for cookies, we normally check it at 225 gram per sub. We have a 6 sub, and we have a standard that if average 1 insect fragment per gram will be a violation. So that's standard there. And if that insect fragment is not reached the threshold, but rodent or mouse hair will also be concerned, because if you find a rodent or mouse hair, you mean what? In this unsanitation situation for the food manufacturer, and maybe the mouse will have pellets in there. That's why the hair come from.
Right.
So the food will contaminate. We will find three. If we find 3 mouse or rodent hair, and in the 3 sub, then will be subject to violation. But of course, that the standard will be changed. But most of them will be stable in there. Like right now, current defect action level, we are there almost over 20, 30 years, not changed. But different food, but some food doesn't have that defect action level. So then we will be analysts. We said, okay, we give it class 2. Then we have a center called the Center of Food Safety and Nutrition, we call it CFSAN. Over there, then will get a second review. Said, okay, then we have much more big pictures than our lab. So then will be said, okay, according to them information, them lower class from lab class to class 1, it means it's good for the customer, can go to the market. Then also can elevate that from lab class 2, go to class 3. It means a violation. It cannot go into the market. So most if you don't have a defect action level, so will be center review. That's much more. We also have a compliance officer, to make the decision. Yeah.
Right. I mean, it's interesting you're saying that the thresholds haven't changed in 30 years, although clearly microscopes have gotten more sophisticated and people have established standard operating procedures, and it's probably much more effective. So I guess I'm not seeing any other hands up, and so I'm going to have to ask. As you know, there is a move towards starting to use insects as food.
Exactly. Yeah.
So I have, what is your opinion on this and how it's going to be complicating your procedures, right? I mean, if insects are now part of the food, finding them would not be surprising. So, but they're good. That would be good insects as opposed to all these other insects that would indicate a problem in the facilities, right?
Yes, like I think like 2 years ago, I go to International Food Safety Protection meeting. That will have a section about the insect as a food. So
Right.
on the meeting, we even sell that cricket chips, so you can eat that. That's the topic. We are not addressed that yet because it's not common food so far. I guess if the food come in, we'll be said, okay, what kind of species is there?
Right.
So except this species, other species supposed not there. Then if supposed not there, you are there, then that's filth elements.
Right. So this is where the image analysis that you actually can identify to species, not just saying there's chitin. Yeah, there's chitin or there's keratin, but what species is it? Okay. I guess one thing I would like you to, if you don't mind, we have a few minutes. What got you interested in working in this particular? Some of the students taking the course don't have some of the big background. So if you don't mind, I mean, some of them are, they're budding into entomologists. Could you tell us a little bit? I mean, Dr. Hamilton sort of gave us a sort of a summary, but what got you interested in this topic?
Yeah, that's before I'm working on the insect electrophysiology, on how the insects smell. That's why I work in Germany, Max Planck. The Dr. Schneider, Kaiserlin, them developed the EAG technique and single cell recording. Very beginning, I'm very interested with that. So I go to his lab, started twice for almost 1 year. Then I working on the University of Idaho as how to the virus, if it in fact if it carry virus and affect the host quality and all that. After a while, I think like all they doing the research, it doesn't connect with your daily life. So it's a for future. Then I said, okay, well, I need to do something with related with reality, real world. So then FDA opened this position, I just apply. So right now I'm pretty happy because my daily work do affect people's life. If the bad food come to the market, it's not good. Right. So it looked like this work is very important. I really enjoyed it, working on FDA so far.
Thank you.
So I just feeling like I go to a grocery store. Say, oh yeah, this one I check it, it's good. And some food I said, oh, I just want to check it. It's okay, but not that good. So I know something.
Oh, I'm sorry, it's 12 o'clock. I'm going to give those that are not taking the course the opportunity to thank you, Dr. Din, really appreciate it, and give them the choice to leave. And those students that are taking the course, if you can hang out a little bit. I mean, we've had quite a few questions, but I want to give you opportunity to ask any additional. So we have about 10 students. And so let's just give it a second here. How many do we have? I think, George, are we good here? We can just go ahead. And so those of you, if you have any questions, please go ahead and raise your hand and we'll answer any additional questions. Thank you. All right, so people are turning on their cameras. Trying to see everybody here. Okay, so Zoe has a question.